This is, I believe, my third Meet the Scientist. This is actually a variation on the theme Meet the New Scientist. And we are very pleased to have with us this evening Dr. Estefania Quesada-Masachs, who is one of the newest arrivals at the Institute. She is somebody that we're thrilled to have. She's an extraordinary clinician scientist who I am certain will make a significant difference in the life of this institute. So she has joined us this evening to talk about her work, and we'll jump in very soon. We'll carve out some time for some Q&A, which is, I think, some of the best part of these sessions. And I think when it's all said and done, we'll know a lot more about the outstanding work that Dr. Quesada-Masachs is doing, and so let's jump in. Just before we do that, just a little housekeeping. So we'll have a little Q&A up front. I'll ask Dr. Quesada-Masachs some questions just to get the ball rolling, and then she'll have a presentation. After that, we'll have some Q&A. So I would encourage everybody to actually use the chat function in Zoom to ask questions as the program's happening, because we'll gather those questions up and we'll use them for the basis of our Q&A. So with that, briefly, I think you've all seen Dr. Quesada-Masachs's bio, but I just wanted to say that she received her MD degree at the University of Barcelona and completed a residency in rheumatology and a 2-year training program in pediatric rheumatology at Vall d'Hebron University in Barcelona, where she worked as a consultant until 2018. She did a fellowship at the Istituto Gianni Gaslini. I'm probably butchering that. My Italian is not very good. A center of excellence in pediatric rheumatology, according to the European League Against Rheumatism. Dr. Quesada-Masachs has completed her PhD in pediatric rheumatology at the Autonomous University of Barcelona with a cum laude and the mention as an international doctor. So her PhD thesis was focused on identifying immunological differences in relation to age and treatment in patients with juvenile idiopathic arthritis. Following her PhD, she joined Professor von Harth, our new scientific director, when he was serving at the La Jolla Institute for Immunology. And she joined him as a postdoctoral fellow, where she focused on studying the role that different immune cells, cytokines, and other signaling molecules played in disease initiation and progression in autoimmune type 1 diabetes. So Dr. Quesada-Masachs followed Dr. Matthias von Harth here. They have a long, productive working relationship, and that relationship continues here at the DRI. So with that, Dr. Quesada-Masachs, are you ready to jump in?
Yes, I am.
Good. Let me just start with a couple of questions. So what is your personal connection to diabetes? Why did you become interested in the science of diabetes?
Yes. So actually, my personal connection, it doesn't come from like the personal experience of having diabetes or type 1 diabetes in myself or people in my family. It comes from the fact that I wanted to solve an autoimmune disease. So autoimmune diseases are like a puzzle. We still don't really cure them. And I'm a rheumatologist. I have studied in my degree and then experience in clinical practice how we can do a lot to improve the quality of life of the patients. But very rarely we are able to reverse the processes. And I would like to cure an autoimmune disease in this lifetime. To be honest, this is what I really would like to do. And I figure that one disease that is located in a gland, in just one organ, right? Not distributed in the whole body, probably has a better chance to find an effective approach than diseases that are much more systemic, much more spread.
So that's good stuff. And you know, you're plugged into, I think, some of the most exciting progress that's taking place in the field right now. You know, you're kind of embedded in this work. What excites you most about your research, and what excites you about the landscape of cure-focused diabetes research?
I think that so I'm really excited about many different aspects of my research and the research of other of my colleagues, to be honest. So the research itself, it's something that excites me a lot, because we keep answering questions that are important. And contrarily to what we can think every time we answer a question, instead of, you know, like having like a clearer path, it's like it opens the path, because it's kind of a complicated disease. So every time we answer a question, we have like 3 more questions to answer. And this is what we have keep experiencing in the last years. But the reason why we are experiencing that is because we are living really exciting times in terms of technology. Actually, I'm gonna talk a bit about that in my presentation today. We can do so much more. We can answer so much more. We can understand better like the bigger picture of what is going on, and things that were thought to be impossible just 10 years ago are possible today in terms of the science part, right? Like how we study the tissue, for instance, or how we studied it 20 years ago. We still can do the things that we did 20 years ago, but now we can do so much more if we choose to, right? So I'm very excited about how we are in a very special moment in time where we can get so much more information, where we can really process so much more, where we have also many things have changed at the same time, right? Technology has evolved, the instruments that we have at our disposal, how we analyze the data that we can receive, how we interact with each other, like communicating with a scientist who is located in Australia. It's just an email. We don't even need to use the phone anymore, right? Just because of how easy it's to collaborate or to get information from other colleagues, and how much we can get from every single technique. I think that we are living very, very exciting times, and we have many more therapies and targets available nowadays in terms of biological therapies that are very specific, that they started being present in the market like about more or less, give or take, 30 years ago, but they have not been around forever. And well, I think that the times that we are living are really, really exciting.
No question about it. You know, and before we turn to your presentation, I just wanted to add that a number of my colleagues and I went to the Institute to spend a little time with Dr. Quesada-Masachs, and we had just a phenomenal session with her. About 2 hours. We went over time. We learned a lot, Estefania, in that session. We learned an awful lot about your work, its implications for care and potential new treatments for diabetes. And it just, for us, it was just a great reminder of, you know, why we do this work and what an exciting time it is for the field. So with that, please, why don't you go ahead and jump in with your presentation at your leisure, and then we'll again carve out some time for some Q&A when you're finished.
Sure. So I'm going to share the PowerPoint, if that is okay. Can you see my screen?
Yes.
Nice. Hold on 1 millisecond, because this is, I minimize it. Great. Very nice. And do you see my mouse?
Yes.
Perfect. So I'm gonna jump into it. So, well, thank you everybody for being here. I'm going to briefly talk a bit about my professional career. Actually, Michael did a very nice introduction, but I just have a slide about that that I'm going to pass pretty briefly. And then I'm going to talk about type 1 diabetes and the research that I'm particularly conducting. So I was born and raised in Barcelona. I studied medicine in the University of Barcelona. I did my specialty in rheumatology and pediatric rheumatology at Vall d'Hebron University Hospital. And I enrolled into the PhD program where I studied the differences, the immunological differences that adults and kids affected by juvenile idiopathic arthritis were experiencing in response to anti-TNF therapy. Anti-TNF therapy is a type of biological therapy that is currently commonly prescribed in patients with inflammatory chronic arthritis. And because this was an international PhD program, I had a 6-month fellowship at the Gaslini Hospital. The Gaslini Hospital in Genova in Italy is the European Center of Excellence in Pediatric Rheumatology, and it was a really great experience. And then I came back to Barcelona, and I was really happy with my clinical practice. I visited hundreds of patients. I treated them with different immunotherapies that are available nowadays, and I was really happy with my clinical practice. But in terms of the type of basic science that I could, and when I say basic, I mean well lab, right? Basic, not for simple, but like well lab science with all the technologies that are a bit farther away from the patient and more connected to the cells and these more trickier technologies. So the type of science that I had access to, I was not fully satisfied with it. So I figured that if I wanted to solve one of the issues with at least one autoimmune disease, I would rather go and learn from the best. And the best person doing type 1 diabetes research in the world in 2018 was Matías Bonerra, who was at La Jolla Institute for Immunology. And I was really grateful and blessed because he hired me as a postdoc in 2018. Our collaboration our work together went really great. And then I was promoted to instructor in the institution, that is like a semi-independent position. And a couple of months ago, I joined the Diabetes Research Institute. And the Diabetes Research Institute is, as many of you probably are aware of it, a very special place, because here I can connect all my clinical experience, all my clinical expertise with my science expertise. And this is nothing that can really happen everywhere. This is a special place where we have scientists, where we have clinicians, where we have all the variety of individuals and options and tools to really answer translational and complex questions. That is what we really have remaining in the field. As you probably are aware, type 1 diabetes is an autoimmune disease. The insulin pancreatic beta cells get destroyed in the pancreas, or get dysfunctional, or get attacked by the immune system to a certain extent. And it's currently affecting 1.7 million Americans. And this is like more than 300,000 children and adolescents nowadays. And these numbers are rising worldwide, are rising also in the US. When I started really focusing just in the study of type 1 diabetes as a disease in 2018, I think that the number of children and adolescents back then was 187,000. So the numbers are going up at a rate that is not really proportional to the rate of the number of the population. And this is not just happening with diabetes, it's actually happening with other autoimmune disease. And it's something that we need to be aware of, and we really need to study. So type 1 diabetes, I mentioned before that is an organ-specific disease. This means that the cells that are affected are located in one organ, in the pancreas, and those are the beta cells. This is the image of an islet. In green, you can see the insulin, in the insulin-producing beta cells, and there are some red cells expressing HLA class 2, in this case immune cells. And basically in this disease, the pancreatic beta cells get attacked and destroyed to the immune system, and then they get also dysfunctional, whatever is remaining, which means that the patient is, this leads for the patients to be needing insulin and control during their lifetime, because currently, these are still the best therapies that we have to keep the patient under control nowadays. But this causes a burden, of course, even though the therapies have been evolving and improving a lot. And the reason why we still don't cure the disease is because we have many gaps of knowledge. So every time we are learning more and more about the events that are going on in the pancreas of individuals with type 1 diabetes and how these differentiate from the healthy, from what we see in the healthy pancreases. But the questions that we need to reply are complex. And we need to fill these gaps in order to be able to really find effective therapies to prevent and to treat the disease. And probably what these therapies won't look the same for patients in different stages of the disease. So we really need to fill the gaps to find these effective approaches. So in my research on type 1 diabetes, there are a couple of things that I wanted to stress. I decided to focus in the study of human type 1 diabetes. Why was for me important to study humans? First of all, I'm a medical doctor, right? So my background, my strength is in the human, in the fact that I participated in many clinical studies and clinical trials. I have the clinical experience of visiting patients, of prescribing immunotherapies, and combining that with the scientific therapy. So for me, it made more sense to really study the human processes. Also, we know that in the animal models, we have many animal models. The animal models that we commonly use are mice. We have nowadays more than 300 therapies that cure mice, but those therapies are not that effective in humans. I believe that the mice models have a place in research, and this place is relevant and is important, and we have learned a lot from them. But when I had to decide where I was going to focus my personal efforts, I decided that it was better to focus on the human aspect of the disease and the human the human-like models that I had available. So what I use is I use cutting-edge technology and innovative approaches to study human pancreatic tissue sections, and I use 3D models that are in vitro models that are studied in the lab, where I can replicate the islet immune interactions, and I can replicate some of the events that take place in the disease. And I'm gonna expand later on this technology. So my objective overall is to understand this interface of communication between beta cells and the immune system, and understand how the tissue microenvironment is influencing the outcome of those interactions, what makes one interaction to be positive and another one to be deleterious, and also to identify those immune events that take place in disease initiation and progression, and who is driving the rest of the response once these immune events happen. And coming back to the technology, why is important to me to use new technology? First of all, with new technology nowadays, when we study the tissue, we can analyze more targets at the same time, and with this, we minimize the use of the precious pancreatic tissue. Here, for instance, I'm showing one image from one of our previous studies where we analyzed the presence of IL-6. IL-6 is a pro-inflammatory cytokine. We have therapies to block it, and at some point we thought could be involved in the disease because apparently beta cells and alpha cells can secrete it. So here you can see an islet, a human pancreatic islet stained with IL-6, so showing this disposition of IL-6 in the islet, of the insulin in the beta cells, and of the glucagon in the alpha cells and the merge signal. And traditionally, we were able to see these 3-4 markers, 3-4 proteins in the tissue at the same time. So currently at the DRI, we purchased this instrument just a few months before I joined, where we can study many more proteins, many more targets at the same time. And why is this important? Because here I'm not able to answer just one question at a time. I can answer many questions simultaneously and get a better big picture landscape of what is really going on in the tissue. Here, for instance, this is tonsils, tonsil tissue of a human donor. And tonsils are very, very rich in immune cells. And here you can see staining for different immune cells. For CD4 T cells, these are one type of immune cells, CD8 T cells, another type of immune cells, CD20 B cells, other type of immune cells, CD45 as an overall, CD68 and 163 as marker for different macrophages, FOXP3 for regulatory T cells. So basically, we can get a lot of information of the different immune cells, in this case in tonsils, but of course we do that in pancreas. And why is it important to study so many markers for me at the same time? Because to give some perspective, if we use the previous system that we have access to, studying 3-4 markers at the same time, if I really want to study 20 proteins at the same time, I have 2 options. I can use consecutive slides, or I can go through multiple cycles of processing the tissue, removing the fluorescent signal, processing it again. And basically, if I use consecutive slides, I'm going to use a large amount of tissue. And the cells that I'm seeing in the first slide are not going to be the same cells that I'm seeing in the last slides, because cells are really tiny. And in every slice of pancreas, I'm losing some of them. So then I cannot understand anymore how these cells are interacting with these others, and how this is affecting the whole picture. Maybe for the first 2 or 3 is fine, but then the farther away I am, the less connections I can make. And if we go through multiple cycles of staining and restaining, we can, of course, see what is going on in the same tissue section, but unfortunately, after a couple of cycles, we start losing some of the tissue and getting some deterioration, and then the results are not that robust anymore, and we see artifacts and other issues that really complicate the story. So it's important to choose a method that where I can see a lot of targets, but with really good quality, using as little tissue as possible, and that provides results that are robust and that more reproducible. So here, this is just an image to show you how it looks with previous microscopy systems that we use, and the quality is really good. We can see the insulin identifying the beta cells, and HLA class 2 in red, in this case, identifying immune cells, and some of these beta cells are also expressing low-key HLA class 2. And here we can see with the new system, many of the immune cells identified at a time. Actually here, I'm providing more details of these immune cells, just showing the colors 2 at a time. And here we can identify CD4 T cells and differentiate them from CD8 T cells, and differentiate them from macrophages and from overall CD45. So the quality, you can see that visually it looks nice, but when we see the metadata, the good thing is like there is what we call a very good signal to noise ratio. This means that whatever is the real signal is very different than whatever is the negative no signal. And we really need this in order to do the type of studies that I'm going to show you in a second. And these studies that I do with the tissue, so I study more proteins than we did before with this new technology, and also apply more complex analysis. And with this, I maximize what I can learn and how much I can learn from every single tissue section. So traditionally, we had, this is one pancreatic section of a pancreatic donor, and every single small green dot that you can see in the tissue is an islet. This pancreas probably has 300, 400, 500 islets. Traditionally, we studied 30. And we selected those islets from different parts of the tissue, and we tried to get a bit of the feel of everywhere, but there were lobules that sometimes we could not get an islet from, or they didn't get selected. So with this system, we studied a limited number of islets at a time, maybe 10% of them, maybe even a bit less than that. The results were reproducible. This was studied by Matthias Monerrat's lab, and his team checked actually that we can we can select 30 islets, and other teams have did that afterwards, by the way. And with 30 islets, you get reproducible results. But there are some risks. There is the risk of a selection bias, even if it's involuntary. There is the risk of totally missing the outliers. Maybe we get what is going on in general, and it's representative of the overall of the tissue, but we may lose what is going on in specific lobules. And we know that type 1 diabetes is pretty heterogeneous. And in this lobule, we can have islets that look great and they seem 100% functional. And then in this lobule, maybe there has been complete annihilation of those islets and they are not there anymore. And then in this lobule, we are getting a strong immune infiltration and the fight is in process. So we really get a lot of heterogeneity, and we can lose that. It will be difficult for us to grasp subtle differences and what is going on with like these outlier islets. And we learn a lot from outliers in clinical and sometimes also in science. So I was not really satisfied with this, so I developed a system using machine learning to study the whole tissue. So, excuse me. Sorry. So machine learning is a type of artificial intelligence, but it's not like ChatGPT. It's not like some very trendy things that are going on online. In this case, I use machine learning using specific software. It's a software that was designed precisely to do image analysis in microscopy tissues. And what I do is like I train the system. I train the system to identify every islet. No? So I basically tell it, I don't train every islet. So I tell the system to identify the islets. I tell the system, this is an islet, this is not. This part is also an islet, this part is not. And I keep feeding it data. And then at some point, the system is able to tell me, okay, these are islets and the others are not. And I do the same for the tissue. So actually the purple outline is what the system defined as the tissue after the training. And here you can see an example of one of the islets being identified and classified by the system after the training. Then once you have them classified, I can do other things. For instance, I can create these peri-islet regions to understand what is going on, not just in the islet, but close to the islet, because the proximity of the islet is very important. This creates this microenvironment. And then once I have these regions, I have the cells, and I can identify, I can ask the system to identify them separately and to classify them based in the presence of the different proteins that I'm studying in that particular tissue section. So this classification, I don't do it just in the islets, I do it in the whole tissue. So once the system is able to identify the whole pancreatic tissue section, I ask the system based on the nuclear signal to identify every single cell. There are different strategies to do that. I use a couple of them, and in this case was based in the nuclear signal. And then it classifies after the system identifies every single cell, then it classifies them based on the presence and the positivity for 1, 2, 3, 20 of the markers that I'm studying, which means that I can have different subclassifications of the cells to understand what are these immune cells, how active are they trying to protect the islet, are they trying to destroy the islet, what is going on with the beta cells, are they stressed, are they happy? That's the reason why the classification matters in those cases. And besides that, I use other software, also machine learning software. Again, it's artificial intelligence, but it's not some of the fancy stuff that you see outside. This is more like these are software, again, designed for image analysis, where I feed those unsupervised systems with all the cellular data, with the data of every single cell. And without training, I ask the system, tell me how many cell types do you see? Every single cell will be classified as positive or negative for 1, 2, 3, 20 of the markers, right? And this is a very beautiful picture where there was this panel very rich in immune cells. And this is tonsils. It's actually a tonsil tissue of the same patient I showed you before in the previous slide. And here you can see these green structures. Every single green structure is actually a germinal center. So the system was able to provide the microanatomy of the tissue because of the proteins that were expressed in the different regions. And it was actually very accurate. So it's a great system to identify microanatomy and to see how is the correlation and the relation of these different cell types without training, like in an unbiased way. So it doesn't matter what I believe, the system will do what it sees based on mathematical equations, that these are the things I need to program and choose, right? The mathematical equations. And then with this unsupervised analysis, I can understand other things. I can understand how beta cells are connecting with immune cells in a pairwise type of interaction. And I can understand neighborhoods. Neighborhoods is like a new concept in tissue, well, relatively new concept in the tissue study, where I basically understand like a small functional unit. So the aim of studying neighborhoods is to understand a very small functional unit where cells are interacting with each other in a particular way, and this is getting to some outcome. And some neighborhoods are telling us or are more frequent in the disease. Some neighborhoods are more frequent in the health pancreas. And here, for instance, just to give you an example of how this works, if one of these cells is really mean, right? Like you have a criminal in the neighborhood and it's really secreting all of these inflammatory cytokines, and it's bothering and it's creating more inflammation and more disruption. You will have a neighborhood probably quite dysfunctional, and this is one of the things that you can observe in disease. But if all the cells are in equilibrium and they are interacting like in a neutral way, kind of with each other, then you would have like some sort of a healthy neighborhood, right? When people are working together and collaborating together, but not really, you don't have a criminal there trying to do something mean, and then another one installing a video vigilance camera, and another one protecting itself with a machine gun, and then things happen, right? So actually, this is just to give you like a visual, right, of how these cellular neighbors work. They are functional units that interact with each other, and it's important not just the unit itself, but also how a neighborhood interacts with the next neighborhood and the next neighborhood, and how this creates these like waves, right, of reaction. And just to give you like the visual perspective of how this has this type of technology, applying this method, applying this new technology that we have available here at the DRI has changed how much we can learn. I did a pilot test with 4 pancreatic donors, 1 non-diabetic and 3 diabetic. And typically, I studied and stained the whole pancreatic section with these 20 markers. And typically, I would have learned from 4 proteins, and I would have selected 120 islets and get the data from a few hundred cells. But in this case, I generated data through this process of more than 1,000 islets, and I got that data from more than 5 million cells. And I could get context of this data and about how these cells were interacting with each other, which means that the system in this case identified 9 cellular clusters, and they had different specific phenotypes like characteristics of these cells, and the cellular neighborhoods that were more abundant in the non-diabetic were not necessarily the ones more abundant in the diabetic, which means that there were different functional units based on the disease status. And coming back to type 1 diabetes per se, again, it's an autoimmune disease. Insulin-producing beta cells are attacked by the immune system, but it's not linear as a disease, right? So we know that there is this, we have this beta cell mass, and we have pre-diabetic individuals who are autoantibody positives, which means that in these patients, we can start detecting autoantibodies in blood. This would be a good moment for intervention, right? If we want to do a prevention study, we have individuals that are at risk of developing diabetes, or according to the new consensus, like already developing diabetes, but in a very, very early stage. Then we have the abnormal oral glucose tolerance test. This is also a good, great moment for intervention if we wanna go in a place where the beta cell mass and the beta cell function will be fluctuating, but still will be really strong. And another good moment for intervention would be just like the diagnosis. We know that it's very common to have the honeymoon phase where the patient is still not needing insulin for some time, but sometimes it's a relatively long time, which means that there is still some beta cell mass and some beta cell functionality that can be preserved many times. So we know that it's an autoimmune disease, but it's fluctuating, it's not linear, and there are persisting beta cells, even in patients with type 1 diabetes that have been having the disease for a really long time. And some of them may not be very efficient at secreting insulin, but insulin is visible in many of the stainings when we analyze the pancreas. We know that the degree of immune infiltration is not huge. When we see the islets of the mice affected with autoimmune diabetes, they really have very strong hundreds of cells involving and destroying the islet. In humans, we see something more subtle, something like what I showed you in the previous pictures, where there were a lot of immune cells in that islet. This was a high degree of infiltration for a human. Sometimes we see 15 cells in an islet, and many immune cell types are involved, and different scientists have suggested that they play a role macrophages play a role. Probably many studies go in that direction, showing that CD8 T cells are the most abundant immune cell infiltrate. CD4 T cells are also present. Dendritic cells can play a role, which means that many immune cell types are playing a role, but we still don't know what is really driving this inflammation. We know that the exocrine pancreas is also involved in type 1 diabetes, and one of the reasons why we think about that is the case is because we see infiltration by immune cells in the pancreas, in the exocrine pancreas of patients with diabetes more commonly than what we see in the healthy controls. So they also have certain level of infiltration, but it's less, it's a bit less intense. And we have seen that in most of the studies where they have where this exocrine involvement have been explored. And also we have these studies observing this smaller size, physical size of the pancreas in the patients with diabetes compared with the controls. So the endocrine units, the islets, are really small. If the pancreas is macroscopically smaller, this necessarily means that, at least to a certain extent, there is some involvement of the exocrine pancreas, so, some of it. And there are some features of the tissue that are typical and characteristic of type 1 diabetes, like the hyperexpression of HLA class 1, that has been described by many scientists. And basically, this hyperexpression of HLA class 1 means that these beta cells will be visible by CD8 T cells, and prospectively, that are one of the, what we believe are one of the main guilties, probably, in the immune attack. And then we also recently confirmed, described along with other colleagues, that HLA class 2 can be present also in the beta cells. This means that in principle, beta cells maybe are able to also communicate with the CD4 T cells. And I say maybe because this is a gap of knowledge that that we are trying to solve. And what I'm studying, one of the things that I'm studying is this islet immune interface, right? I know that beta cells can hyperexpress HLA class 1. This would mean that it may make them visible and susceptible to be attacked by antigen-specific CD8 T cells, that these are basically cells that are programmed to go and kill the beta cell. The interesting thing is, like, when my colleagues and especially Dr. Van der Rath investigated the presence of these cells, they observed them that they are also present in the healthy controls. It shows that the numbers are lower and they are mostly in the exocrine. They are less frequently close to the islet, but they are there, right? Which means that not because they are there, they necessarily need to attack. So this is also really interesting to me. And also beta cells, I mentioned, that can express HLA class 2. This means that can make them visible to CD4 T cells. But if CD4 T cells sees them, actually, depending on the other co-stimulatory molecules and other things that are going with this stimulus, this can start a deleterious response where immune cells respond in an inflammatory way and try and participate also in this beta cell destruction, or they can respond in a protective way, having more of a regulatory, like pacificator type of response, trying to slow down the inflammation that can be around them. And to answer those questions about how these beta cells are interacting with the immune system in a dynamic way, because when we study the pancreas, we have a snapshot, right? We know what was going on in that moment. We can interfere pseudo timelines, which means, like, we can more or less assume what was going on at different points, before the attack, after the attack, etc. But we can recreate some of these processes in vitro with these also very novel assays, like the one with islet spheroids. And islet spheroids are created from the islet cells of human pancreatic donors. And basically, we create these. These are generated in Switzerland. This is specifically that I'm showing here, where we get these plates, and every single well has like a perfect islet. And the reason why this is interesting to use is because the responses between them are very similar. So if you do an intervention in one, you have a similar response to the other. And why is this important? Because the native islets, our islets, when they are just isolated from a pancreas, when a patient donates the organ, we don't get equal islets. We get all over the place islets. Some of them are really small. Some of them are really large. Some of them are intermediate. Some of them have a strong functionality. Some of them are a bit less functional. And when we are comparing the effect of an intervention in vitro in the lab, it's difficult sometimes to compare the responses. We have our ways. We have optimized how to get like more comparable results. But in principle, with the islet spheroids, we get results that are a bit more robust, and this is important for our study. So basically, we can co-culture these islets with these human islets and human-derived islet spheroids with human immune cells, and do different things on them. For instance, stress the beta cells, make them express HLA class 1, make them express HLA class 2, and see how they react and how they interact with the immune system under different conditions. And then we can see how the islet function changes. What means is like how the secretion of insulin changes, how the C-peptide secretion changes. We can do confocal microscopy to study what proteins are being expressed now, and if there are immune cells around, are they infiltrating, and how much are they infiltrating, and are they in close contact of the beta cells that are more stressed, or are they just randomly infiltrating? And then we can do flow cytometry to answer questions about specific characteristics of those cells, or transcriptomic studies, again, to answer more genetic, broader characteristics of those cells. And this is a video of one of these islet organoids. In green, you can see the insulin. In blue, you can see the HLA class 2. And in purple, you can see immune T cells that are, in this case, in contact and infiltrating the islet. This islet spheroid was a bit stressed. And with this 3D analysis, we can get like the structure, preserve the structure of the islet, and then we can see the inside of it. We can analyze how many cells were expressing this or this other protein. We can see, we can visualize if the expression, where the islets were located, how the cells were packed, if the expression of HLA class 2, in this case in blue, was being connected with the presence of these T cells, if they were communicating, or if the location had nothing to do with it. And when we do this type of analysis, you will see in a minute that we don't necessarily work with the whole picture. For instance, here I was just interested in the cells that were touching the islet or already penetrating the islet, but we had a lot of other T cells, as you can see here in the original signal, around the islet, that because they were not interacting, I assume were not that relevant to understand how was this direct communication between them.
Very impressive graphics. Just a real quick time check. I want to make sure we have plenty of time for Q&A.
Yeah, thank you. Sorry, because I cannot see.
Not at all.
Yeah, so basically, just to finalize, we know that many of the immune cells are involved in type 1 diabetes. We still don't know who is driving this immune response against these pancreatic beta cells. And with these technologies and with these systems, we are getting a better understanding of the full picture of that, and hopefully we will be able to solve some really important questions that we still have pending. And thank you very much.
Really good stuff. Thank you so much for that presentation. Well done. And I wanted to make sure we had plenty of time for Q&A. I have a question. I'm not going to ask it though, because I want to defer to our guests. Anybody who wants to jump in with a question, please go ahead. I'm gonna ask mine. Could you talk to us a little more about the importance of machine learning in your research? It is, again, we're not talking about chatbots here, right? But specifically, how are you leveraging machine learning to advance your work?
Yes, so the good thing of machine learning is that it allows you to automatize or to semi-automatize some of the processes. So when we, as humans, need to choose, right, what cell is expressing a lot, what cell is expressing little, based on our perception, we commit a lot of errors, right? Because some days we are tired, some days we are having a better judgment, some days we are using as reference X, and some days we are using as reference Y. So the good thing of machine learning is that it kind of homogenizes this type of studies. You still need to, in the first set of things that I showed, you still need to train it, which means that the system doesn't necessarily has answers at all right away. You need to tell this is X, this is Y, this is nothing. And then it gets results. And the system is not perfect. Sometimes the systems get creative. For instance, if you overtrain it, if you feed it with too much information, at some point it gets a bit dumb. It doesn't really know what is doing anymore, right? So there are sweet spots and there are SOPs actually, right, that we need to follow in order to get like the best out of it. But the reason why it's important to use these semi-automatized processes is because we cannot, as humans, count 5 million cells just out of 4 patients. If we want to get all the information from every single section, and I think that this is also there. We can do it, right? So in my opinion, is the fair thing to do for the donor themselves, right? They donated the pancreas to learn as much as possible about the disease. So we need to do that if we can, because we have the means, we have the systems, we have the software. It just takes a bit of time to train the software to be smart, but yeah.
Thank you for that. And we do have a question from via chat. How would increased funding impact your research? How would you allocate the money? What more would you be able to do?
Yes, so thank you for the question. So for instance, with more funding, we were able to purchase the machine. This, the DRI purchased it, as I mentioned a few months ago. These technologies, unfortunately, not cheap, right? So the system itself, I don't want to say a number that is inaccurate, but it's something surrounding the $500,000 just the system. And then you still need to have to receive technical support. You still need to have 2 scientists, typically a senior and a junior, that will be devoted to make sure that the quality control and everything is good with the system. And this is just for the first few months. And then you need to have at least collaborating with you engineers. And again, engineers are also, they are a really great resource, and they have a value that you need to pay for if you really want to have good engineers collaborating with you. And we need that because these systems work with Groovy scripting, or you need to do programming sometimes, like low-key programming, but still programming. And I mean, I'm a medical doctor. I can do some of it. But of course, if you want to have the best outcome, you need to have the best collaborating with you, because you cannot be the best in absolutely every single thing, right? So yeah, so when you have more resources, you can hire better collaborators, and for longer periods of times, you can incorporate them in your teams. And also the antibodies that we use. So in the past, when we were doing a staining, we were studying 3 proteins. This means we were buying 3 antibodies. Every antibody can have an average cost of $500. With every single vial, I can study maybe 20 to 50 samples. It depends, right? This is more like general. But now I need 20 antibodies, and they still have the same cost. Actually, the costs have gone really high, to be honest. So if I have more funding, I can explore different proteins. I can ask different questions. I don't need to just do one panel of these 20 proteins and then forget about doing another panel in 1 year, right? I can just go right away and do another panel. So with this, these are things that are cost to consider with this newer technology.
So we have another question from Deva. So, and another one one from Bob. So a wonderful presentation, Estefania. Thank you. I'm wondering if this incredible technology and your research will also benefit type 2 diabetes as you focus your cure-focused research on type 1.
Yeah, this is actually a really great question. So yeah, I have to say that most of my focus is on type 1, but I also study the pancreatic tissue of patients affected with type 2 diabetes, because the more we learn, the more we understand that type 2 is not just a metabolic disease. And I'm saying that it's really complex metabolic disease. I'm not saying just as this is easy and we can solve it however, no. But on top of that, we know that the immune system is playing a role there. And that role may be more important than we initially thought when we didn't have access to the tissues. But for instance, when we did our study, in IL-17, that is one pro-inflammatory protein that relatively surprisingly, but beta cells and alpha cells apparently are able to secrete it. And relatively surprisingly, because these are a type of pro-inflammatory proteins that are typically secreted by the immune system or to connect with the immune system, which means that there is this at least type of indirect communication. This IL-17 that is inflammatory, it was not just present in the islets of patients with type 1 diabetes, it was also present in the islets of patients with type 2 diabetes, which means that there is at least this low-key inflammation there that for sure is playing a role. And other colleagues that are studying the presence of these like macrophages and other immune cells in the fatty tissue of patients with type 2 diabetes, they are seeing also these patterns of inflammation that are actually really interesting and not what we initially thought about. So yeah, to me, from the immunological perspective, type 2 diabetes is also interesting. It's just that from the genetical perspective and other aspects of it, type 1 diabetes is closer to juvenile idiopathic arthritis from the genetic prevalence, which means that the root or the type of error going there, it's different, but this doesn't mean that the immune system doesn't participate. And we can use definitely these systems to also learn more about type 2.
Thank you for that, Deborah. Thank you for that question. Bob asks, where does the human tissue come from? Are they readily available? How do you see the DRI working with others doing similar work around the world?
Sorry, what was that question? I was
Sure. So
here, Bob.
Human tissue from, yeah. Mm-hmm. And how do you see the DRI working with other collaborators around the world?
Well, we are actually having a lot of collaborations. For instance, the tissue that I'm working with, most of it comes from MPOT. I don't know if you're familiar with MPOT. It's a network of pancreatic organ donors with diabetes. The headquarters is here in Florida. We actively collaborate with them. Me personally, I also collaborate with them. They kind of standardize how the pancreas is processed and it's distributed to researchers. This is important because when the patient dies, the exocrine portion of the pancreas starts digesting itself. So you really need to process the tissue in a way that has really good quality and that is really robust, because otherwise the gland gets kind of destroyed, digested. So it doesn't really have good quality. It doesn't serve for research. You cannot use not just this technology, with any technology won't be good, right? So one thing that really improved so much with MPOT was the fact that they made available really high quality pancreatic tissue to the scientific community in the US, but actually in the world. We have collaborators in other countries. I also have received pancreatic tissue sections from our collaborators from Europe who participate in this DVID study, where I don't know if you're familiar with the DVID study, but basically the pancreas of 6 patients who started having diabetes was biopsied. And these biopsies are really super precious. Those patients are still alive, actually. And this is very rare. Most of the times we work with the pancreases of organ donors, which means individuals who passed and they donated the pancreas and then they were processed for scientific reasons. And we also had access to that. And well, we have collaborators all around the world, actually. At the DRI, for sure, all around the world. And me personally, well, so far some in Spain, so far in northern Europe, across the US, UCSD, for instance. So we have different collaborators.
Thank you. Thanks for that question, Bob. Sophie asks, you mentioned that you didn't get into this research from a personal or family history. I'm wondering, from your experience in this research now, can you share an experience with a diabetes patient that has impacted you?
Yeah, many actually. Many. Some of them come from my colleagues, because I actually have colleagues who are affected by type 1 diabetes, and I work with them in a day-by-day basis. And as a clinician, my patients were affected by chronic inflammatory arthritis. Most of them were children. Some of them were adolescents, some of them were pregnant women, some of them were young individuals, and at the beginning of my career, some of them were older individuals. But I didn't really have, I'm not an endocrinologist, right? So I don't control glucose. So I'm the person to decide the immunotherapy. I'm not the person to decide the insulin, 100%. But I had a lot of experiences with my colleagues and the people in my team who is affected with the disease, by the disease, and I have learned a lot from them, because it's always much more like visual, right? To see the day by day while you live it, than just to read about it or hear about it in a medical visit for like 30 minutes, right?
Thank you for that. Maybe just one more question. I think we're at the end of our time. So real quick, hello, Dr. Estefania. It was a wonderful overview of your research area. I'm curious about the software and how you could use it to analyze 20 different colors if they overlap.
Yes, so this system, I didn't go into the technical details, but this is a really technical, really great technical question. So basically, this system takes the whole spectrum of the light, similar to new flow cytometry systems that are also taking a lot of the spectrum of the light. And you can use fluorochromes that, instead of being very spread as they were like the older previous ones, they have to be more narrow, so they separate better. One color from each other is separating better. But the same thing that happens with, similarly as happens with complex panels in flow cytometry, you need to deconvolute the images and the colors, so to kind of artificially separate them a bit. That's the reason why you need to have really good signal to noise ratio. So you cannot move on with a bad antibody. You cannot move on with a I'm not sure type of signal. You really need to be very confident. You need to have a lot of controls. Actually, the process of having a panel with the system is not easy. It takes months. So you really need to think, what questions do I want to answer? What do I need to answer these questions? So one thing that new technologies are not, at the beginning, at least, are fast, right? Because you need to prepare them better, because more problems can happen along the way. So you need to have everything more structured. So there is the convolution steps where overlap is handled similarly as it happens with flow cytometry panels.
Thank you for that. Well, we're just a couple minutes over, so why don't we wrap up? I want to just end by thanking you. Thank you so much, Dr. Quesada-Masachs, for joining us for an amazing presentation. Welcome to the DRI. It's so good to have you here. I think the Institute is a much stronger place because of your arrival. I want to thank all of the guests who've joined, the questions. I think this is an extraordinary time for the field of diabetes research and care. There's no question about it. The Institute is fortunate to have some of the most talented clinicians, scientists, researchers advancing the field with a focus on the cure and a focus on the patients. And I want to thank everybody who supports that mission, who's on this call. We cannot do it without you. Once again, Estefania, thank you so much for joining us. And unless there's anything else, we'll bring the gavel down on this session.